just say a few words. I'm Bruce Ames from biochemistry. Last time, as you know, Kashan talked a little bit about Pauling's many contributions in many fields, and of course, Dr. Pauling talked about his work. But I was a graduate student at Caltech in the early 50s, and it was a small place, and sort of everyone there knew that if you wanted ideas on your project, the person to go talk to was Pauling. There was just a constant stream of new ideas coming up, and but in very many different fields. And I think that in his contributions, as you all know, have been really enormously diverse for a scientist in so many different fields. And I think in recent years, his intuition is still really fantastic, because the work that he's been doing on metabolic profiling and sort of looking in different people for sampling metabolism and getting at human variability, I think is going to start off and has started off a whole area of people working in that area. And then he's had this intuition about oxidants and damage and the role of oxidants in health and the importance of antioxidants. And again, I think more and more evidence is coming up that that's really an important field. So that I think there's so many different contributions and also so many students he's trained over the years that when I was a graduate student at Caltech, some of the fellow students there were working with Pauling were Alex Rich and Gary Felsenfeld and Martin Karplus. And so there have just been student after student who've really made enormous contributions that Pauling has trained. And so one can ask then, is Pauling always right? And I'd like to just tell one story about a famous German chemist, a predecessor of Dr. Pauling, Emil Fischer, who was one of the great chemists of his age. And over the years, Fischer would be finding out all sorts of things, but every once in a while he'd find out that Abderhalden was wrong about something. Abderhalden was another great German organic chemist of last century. And so this went on for about 3 or 4 times that Fischer proved Abderhalden wrong about something. And then someone asked Fischer, well, is Abderhalden always wrong? And Fischer thought for a moment and said, no, he's not that reliable. And, so I was asking Dan Koshland the other day, well, is Pauling always right? And Koshland thought a moment and said, no, he's not that boring. So for that reason and others, we're very happy to have Dr. Pauling give his second lecture. So.
Thanks. Well, I'm pleased to be here to give a second lecture. The title that I gave for this lecture was Chemical Bonds in Biology. I had thought that I would call it the nature of life. And in fact, I shall talk about the nature of life, especially the molecular basis of life, or at any rate, as I see it, about my own efforts to understand life, living organisms, and to satisfy my own curiosity. And this effort ended quite a long time ago. I shan't talk about recent discoveries, but rather about the basis on which they have been made. I'm astonished that in recent discussions of life, even in biology textbooks, there's usually rather little about what I consider the basis of life. My interest in the problem of the nature of life began in 1939, when I had been in the California Institute of Technology for 7 years. In 1929, the Institute, which had departments of physics and chemistry that were pretty vigorous ones and had mathematicians and engineers, started a division of biology. Thomas Hunt Morgan, who had discovered the gene in 1911 with his students, came from Columbia to be the chairman of this division. And he brought with him his co-discoverers, except for Hermann Muller, who didn't come. He brought Alfred Sturtevant and Bridges, the two young biologists who had been most active with him in developing the theory of the gene. And he also brought other biologists, including Albert Tyler, an embryologist. I soon got interested in what they were doing and spent a good bit of time with Morgan and the others. There are some that I haven't mentioned too. Well, I had been determining the structures of minerals and other inorganic substances up to that time and doing quantum mechanical work, theoretical work on the chemical bond. This X-ray diffraction technique was really a fine one, which I liked. It was really great. With a little bit of luck, you could determine the structure of a crystal in 3 months, say, and get the answer to some interesting problem in structural chemistry. And then, having got that answer, go on to some other question and get another answer. I hope that scientific research is just as exciting now as it was then. I can't believe that it's more exciting. That was a great period. Well, I was beginning to feel that the puzzling questions in the inorganic world, at any rate, were all being answered. And I realized that there were still many puzzling questions. The puzzling question about the nature of life. By 1928, I felt that the problem of the nature of the chemical bond was well on its way to solution. And I wrote in 1929 a paper on the principles determining the structure of the silicates and other minerals. The silicates have complicated formulas, many of them, but it turned out there were some structural principles that enabled one to see why a feldspar, for example, an aluminosilicate feldspar, which often contained sodium or potassium or calcium, had one sodium atom for every aluminum atom and had a ratio of oxygen to aluminum plus silicon of 2, and so on. Well, these principles also permitted one to determine the structure of crystals that were too complicated to permit a straightforward determination by the X-ray diffraction method that led to the determination of the structure of topaz and mica, zunite, for example. I remember how pleased I was at the structure of zunite. It turned out, and the mineralogists weren't sure what its formula was. It turned out that it was Al13Si5O20OH18Cl. This Cl happened to get in there by the chlorine ion, chloride ion, got in, and one could see from the structure. There was one peculiar aluminum atom in the structure. One could see that the peculiarity in the aluminum atom was responsible for there being a chloride ion in the crystal. Well, the problem that about the nature of life, what is it? I learned from the biologists that biological specificity is essentially the major problem that we have to understand if we want to understand life. Biological specificity is the set of characteristics of living organisms or constituents of living organisms of being special or doing something special. Each animal or plant species is special, shows specificity. It differs in some way from all other species of animals. Its outstanding specificity is that of passing on to its progeny, its descendants, its own characteristic nature. How is it possible for two, you know, you were all pretty sophisticated, bored, jaded now, I suppose, by these things. You may not seem interesting to you, but in the old days, we could ask, how is it possible for two human beings to have an offspring that is a human being instead of some other animal, and even an offspring that resembles his parents more than he resembles other human beings? Well, Morgan, Sturtevant, and Bridges and Muller around 1911 had developed the theory of the gene, and the three of them were there in Pasadena. I was always being exposed to talk about genetics, heredity, specificity, and being stimulated to ask myself over and over again, what can the molecules be doing that confers such high specificity on these living organisms? I doubt that I had any idea that I would ever do anything about this, or even that that anyone would have done enough to give me an understanding of it, but I couldn't help thinking about it from time to time. And it's not only the organisms that show this remarkable specificity, but also parts of them. For example, you can take an enzyme out of plant or animal cells and find that they are able in a remarkable way to make a chemical reaction go tens of thousands times faster than it would go in the absence of the enzyme, with the enzyme not itself being changed. And the enzymes are very particular about what chemical reactions they catalyze in this way. I mentioned superoxide dismutase, a stuff that you can buy in the health store to swallow, and it gets digested there, of course, and that's that. But if it's injected into the bloodstream, it might have some value against disease, and of course it is essential for health of all organisms, of cells, by killing the superoxide radicals that get formed there. So that's what superoxide dismutase does. Very, very minute amount of this protein is enough to keep cells almost completely free of superoxide dismutase. It causes destruction of that radical, and it doesn't do anything else. It doesn't do anything to hydroxide radicals or singlet oxygen, or, so far as I know, anything else. There may be another reaction, another substrate, but if there is, I don't. Well, there's specificity of another sort. That's a high specificity. There's specificity of another sort. There are million species of animals. So far as I know, practically every species of animal manufactures a different kind of superoxide dismutase molecule from other species of animals. So that there may be about 1 million kinds, even more, of this particular protein. Well, proteins are big. A standard protein may have 10,000 atoms in it. You can change some of these atoms, replace a few atoms by another group of atoms without interfering with the catalytic power, not disturbing the active site. And perhaps you confer some benefit for the particular animal species by making this change. At any rate, we have specificity in the nature of proteins that are produced by living organisms, characteristic often of the species, with only once in a while the same protein being produced by two different species. Well, there were lots of things I wanted to understand without much hope. How an enzyme is able to catalyze one reaction and not another. How organisms are able to have progeny of the proper kind, and so on. The red protein in the blood, hemoglobin, has interested me for a long time, has a beautiful color. When I was a young graduate student beginning, I saw in the library, in the chemical library in the California Institute of Technology, some big green volumes, which I looked at through curiosity. They were by, I'm not quite sure, Brown and Schuchert or someone like that. These investigators had collected blood from a lot of different animals. They separated the red corpuscles and hemolyzed them and let the hemoglobin crystallize, and then took photographs of these hemoglobin crystals. And the crystals all look different from one animal species to another animal species. All animals manufacture each animal species manufactures its own kind. Human beings manufacture human hemoglobin. Fetal humans manufacture fetal hemoglobin, which is more like fetal calf hemoglobin, say, than adult human hemoglobin. The fetuses of the world are more closely related to one another in some way than they are to the adults of their own species. Well, here, and of course now we know that about 300 different kinds of human hemoglobins are manufactured by adult human beings, these variants of human hemoglobin. So that's another example of biological specificity. Here, well, in 1930, the year after Morgan came, I began work on organic molecules. I'd never had any interest in organic chemistry, but of course the molecules are molecules and they're interesting, so I worked on them by a new technique, electron diffraction of gas molecules, very powerful. We got a lot of results in that way. And at the same time, having felt satisfied with an understanding of the silicate minerals, I began to work on the sulfide minerals. I determined several sulfide structures, but it was quite a chore. I said 3 months. The sulfide structures are apt to be complicated enough to take more than 3 months per structure, and I got impatient. So I applied to the Geological Society of America for a grant from the Penrose Fund and didn't get it. So then I applied to the Rockefeller Foundation, and they gave me $5,000 for an assistant to help me work on the sulfide minerals. And I enjoyed myself with my having an assistant to help me getting some of these structures. And they gave me $10,000 the next year. Warren Weaver there was in charge. And then Warren Weaver said, the Rockefeller Foundation isn't really interested in the sulfide minerals. What we are interested in is biology. So that sank in for a while. And I remembered those beautiful red crystals of hemoglobin. And in fact, I had written a paper on the sigmoid equilibrium constant of hemoglobin with oxygen, a theoretical paper. That was my beginning in the protein field. So I had an idea, and I applied for a grant to study the magnetic properties of hemoglobin. This was 87 years, I think, after the last work had been done. Faraday wrote in his diary, have measured the magnetic susceptibility of old dried blood. Must try recent fluid blood. So we got the grant. And Charles Coriel and I borrowed an old discarded balance from the sophomore analytical lab and drilled a hole in it and hung a tube from it and borrowed a magnet from Mount Wilson Observatory and measured the susceptibility of hemoglobin, venous blood and arterial blood. And to our astonishment, there was a big difference in susceptibility, magnetic properties of venous blood and arterial blood. Very interesting consequences of the magnetic properties, significance for the structure of hemoglobin. Well, you know, perhaps I'd be a great authority on the sulfide minerals if it weren't for Warren Weaver, or if I'd got the grant from the Penrose Fund originally. Well, I remember once when President Truman, in an address, said, why does Dr. Pauling work on the red cells in the blood? Why doesn't he work on the white cells? Well, of course, now we determine the amount of ascorbate in the white cells. It's required for phagocytic activity, so I've got around to that too. Well, Warren Weaver invented a phrase to describe what we were doing. He called it molecular biology. So, in 1936, I gave a grand rounds talk at the Rockefeller Institute for Medical Research in New York about hemoglobin. And after my talk, Karl Landsteiner asked if I would come to his laboratory and talk with him. He was doing very interesting work. He had discovered the blood groups back in 1900, A, B, and O. And he was then making studies of the reactions of antibodies and antigens, homologous antigens or heterologous antigens. And in fact, it was the sort of work that would appeal to a chemist because he would take a chemical off the shelf, something with known structure, simple substance, and couple it to a protein, usually by diazotization of an amino group, and inject this azoprotein into a rabbit and get antibodies which were characteristic of this simple chemical. He would get a solution of protein molecules that would combine specifically with the chemical that he had took off the shelf, something that no rabbit had ever seen before. Well, this was surely interesting to me. Another example of biological specificity. These immune reactions are about as specific as you can get. You can detect a difference between, say, hen egg ovalbumin and duck egg ovalbumin when, up until recently, there were no other ways of showing that these two closely related proteins were, in fact, different from one another. So I thought about, I couldn't answer any of Landsteiner's questions. I thought about them for 4 years, and I think I'll tell you later what came out of my thinking about them. Well, I want to put in an interlude here about chemical bonds in biology. In my book, The Nature of the Chemical Bond, first edition, 1939, I wrote on the last page of the book, among the most interesting problems of science are those of the structure and properties of substances of biological importance. I have little doubt that in this field, resonance and the hydrogen bond are of great significance, and that these two structural features will be found to play an important part in such physiological phenomena as the contraction of muscle and the transmission of impulses along nerves in the brain. Well, at that time, 43 years ago, there was little known, 44, isn't it? It's 1983. 44 years ago, there was little known about the structure of proteins. You may not believe it, you young fellows and young ladies, or women, I guess it's proper to say now. But 44 years ago, there was even some doubt as to whether proteins consisted of polypeptide chains. An English scientist, Dorothy Rynch, had proposed structures for proteins in which there were no polypeptide chains, no double bonds from carbon to oxygen in the carbonyl groups, which instead had opened up and formed single bonds. And these were two-dimensional, well, some three-dimensional, but sort of planar structures bent around. They were very interesting from the geometric or even topological point of view, but of course were wrong. They're just forgotten about now. But back in 1939, Irving Langmuir, the same one that I talked about on Monday, great man, Irving Langmuir wrote a paper with Dorothy Rynch giving arguments as to why these cyclical structures were correct for proteins. And this disturbed a colleague of mine, Carl Neiman, protein chemist, and me enough so that we wrote a paper that was published in 1939 giving the arguments for the polypeptide chains. And of course, they're overwhelming. Ever since the time of Emil Fischer, the father of H.O.L. Fischer, who was here, it's been pretty certain that the polypeptide chains are present. Well, there was then the question. In fact, in 1936, Alfred Mirsky, who worked with me for a year, and I wrote a paper on proteins in which we said that a native protein is held in a well-defined conformation by weak forces between parts of the chain, including and perhaps especially hydrogen bond formation. And many people, Huggins, whom I've talked about, for example, had got interested in the question, also William Astbury in England, the question of how these polypeptide chains got folded. Here you have a long polypeptide chain with 100 or 200 amino acid residues in it, and with carbonyl and NH groups that can form hydrogen bonds with one another. How does this chain fold up in a globular molecule, or how are the chains related to one another and to themselves in a hair, for example, alpha keratin? So by 1937, I had reached the conclusion that I knew enough about bonds and carbon, nitrogen, and hydrogen atoms to be able to find a structure for alpha keratin. I spent the summer of 1937 folding these polypeptide chains. I assumed that the peptide group, the amide group, was planar because of resonance. This is where resonance comes in. And that the chain was folded around in such a way that the CO group and the NH group would form hydrogen bonds with one another. And I also, from the X-ray pictures of hair, there was a well-defined, apparently meridional reflection off the direction of the axis of the hair with a spacing of 5.1 angstrom, which everybody assumed meant that the structure repeated every 5.1 angstrom in that direction. So I spent the summer without finding any structure. And this is what had happened to Astbury, too. He described a structure, but it wasn't right. And so I decided that there was something missing in my knowledge about chemistry, structural chemistry. That perhaps a secret of life was hidden in these protein molecules. Perhaps there's a structural feature, a chemical feature about polypeptide chains that hadn't been discovered from the examination of simpler substances. At the end of that summer, Robert B. Corey came to me from the Rockefeller Institute for Medical Research. He was interested in proteins too, and we decided that the thing to do was to look for this unknown structural feature. Nobody up to 1937 had ever made a structure determination by X-ray analysis of crystals of any amino acid or peptide. And we decided that our program would be to determine the structures of these crystals. It was sort of at the borderline of science then. The techniques of X-ray crystallography were improving, but they were far from what they are today, when it's a pretty routine job to determine the structure of a crystal. It wasn't routine at all then. It would take a year, a man year, or 2 man years. Many people were involved in this work for over a period of years, continuing to the present time. At the end of 11 years, we in Pasadena had determined the structures of a dozen amino acids and several simple peptides. And no one anywhere else in the world had determined any such structure. We were interested in doing it. I looked them over in 1948 and realized that we hadn't made the discovery that we wanted to make. The dimensions, the structures were all just what I had thought they would be back in 1937. There wasn't any secret of life hidden in the folded polypeptide chain, so far as these basic structural characteristics go, the bond lengths and bond angles and so on. And so one day I thought, why don't I think about how to fold the polypeptide chain again? And I just sort of forgot about the evidence, the 5.1 angstrom repeat, and just thought about the bond lengths and the bond angles, the planarity, the resonance in the amide group, and the formation of hydrogen bonds. And in a couple of hours after I had made the decision, I had discovered the alpha helix, without even a molecular model at hand, only a sheet of paper that I could fold in three-dimensional space. And what was wrong in 1937? Paying too much attention to that 5.1 angstrom repeat. It turned out that it isn't a repeat distance after all. It's a sort of pseudo-repeat. Well, I think I'd better start in with those slides, because I have them and I'd feel that I needed to show them, and then it might be too late. Can we turn some lights off too? Well, here I just show the hydrogen bond in diacetic acid, essentially as described qualitatively by Latimer and Rodebush up here in Gilman Hall in 1920. And here is a molecule of glycine as described for the first time about 1916 here in Berkeley by a man named Elliot Quincy Adams. He didn't stay here long. I'm not sure whether he took his doctor's degree. I think he did here. And also at the same time by Bjerrum in Copenhagen. Well, the aspect that they discovered was that one end is the ammonium ion group with a positive charge, and the other end the carboxylate ion group with a negative charge. And here we have two glycines which have condensed together, eliminating a molecule of water, forming the peptide bond. And here we have a lot of amino acids lined up in a chain. And the top way is the way that a chemist looks at it, a series of amino acid residues. The bottom is the way a structural chemist looks at it, a set of planar peptide groups connected by alpha carbon atoms, which happen to have different side chains, any one of 20 different side chains attached to them. And the problem that I had then, in both 1937 and 1948, was to rotate around the two single bonds from carbon to carbon or carbon to nitrogen of the alpha carbon atom that has a hydrogen and another group attached, and find a structure where the CO and the NH would form hydrogen bonds. That was the problem. And of course, alpha helix I found by assuming that all of the structural units are equivalent, and the helix is the most general structure that you get from asymmetric equivalent elements. They always form a helix, which may be a degenerate helix. I didn't worry about restriction of rotation around the single bonds discovered here by Professor Ken Pitzer and Kemp, I guess, back in the 1930s. It's not a big enough effect to bother us as a first approximation. And here's a drawing showing the dimensions. They're within 1/100th of an angstrom or 2 or 3 degrees in angle of the ones that I was using in 1937. I just didn't think hard enough, I guess, in 1937. Here is a drawing which antedates the alpha helix. It just shows what we were working on, a polypeptide chain and folding in such a way that hydrogen bonds are formed. And it shows these space-filling models. Dr. Corey and I were building space-filling models before the alpha helix came along. Now everyone has them. They're commercially available. I remember my son Peter saying that they were the most expensive atoms in the world. So here's the right-handed helix on the right. The left-handed one hasn't been found. There are 4 6/10 residues per turn. So that, and the pitch, the length per turn is 5 4/10 angstroms. That's 6% greater than the 5.1 indicated by the photographs. And it was so far off that it worried me. I knew that the bond lengths and bond angles were so good that it had to be 5.4 angstroms. The photographs looked good, and they said 5.1 angstroms. Well, I delayed publication of this for a year, always expecting that Huggins or Perutz or someone would find the alpha helix in the meantime, because I couldn't understand this discrepancy. You know, no doubt, that I'm a very cautious and reliable scientist. I don't ever publish anything until I'm sure that it is right. So I waited for a year and finally published, Corey Branson and I published this description of the alpha helix. This shows the helix as it goes along with approximately 18 residues in 5 turns, 36. I said 4.6. I should have said, you know, it's remarkable, this reverberatory memory that you have, because when I said 18 residues in 5 turns, I got 3.6, and my memory said, but I've just said 4.6. Well, it's 3.6 residues per turn. So here's one of our models. They were made out of wood, were very heavy, these atoms. That would be a polyglycine with the alpha helix structure, and here with some side chains attached to sort of miscellaneous side chains. And of course, here's that myoglobin picture showing all 2,500 atoms, and you can see off down at the lower left side, that segment of alpha helix. It has 8 segments, 8 alpha helical segments comprising about 85% of all the other residues. Others are involved in bending around the corners. Here's a carboxypeptidase, which has 307 amino acid residues in the polypeptide chain, and only the residues are indicated by little straight pieces of wire. That's, each of them is about 10 atoms. And you can see the several segments of alpha helix in that structure. Well, I was told last month that 100 new protein structures were published last year. Several hundred altogether, 200 or 300 have been published so far. Almost all of them show alpha helix. Some of them only the pleated sheet. Well, here the two pleated sheets that we described, the parallel chain and the anti-parallel chain pleated sheet. If you stretch a hair by taking one of these permanent wave or just steaming it and then stretching it, it'll stretch out to about 2.5 times its original length, and it has the parallel chain pleated sheet structure. But silk fibrillin has the anti-parallel chain structure, which is shown here. The hydrogen bonds are lateral. Even before the alpha helix was discovered and the structure of silk was discovered, it was known that the hydrogen bonds were in the direction of the fiber, the hair, and were lateral in silk because you could determine the anisotropy of absorption of ultraviolet, of infrared light, the carbon-oxygen or nitrogen-hydrogen stretching frequencies, and find out the orientation of the hydrogen bonds. Well, of course, scientists can do a lot fancier things than that now. I continue to be astonished at how penetrating some of the new techniques are. This is a pleated sheet showing the pleats, and sort of from the side. And another view made by my friend Roger Hayward, an architect who had a remarkable understanding of science and mathematics too. Well, it's interesting that nobody discovered the alpha helix in that period between 1937 and 1948, because a number of people were working at it. And in particular, Bragg, Kendrew, and Perutz published in 1948, or after I discovered the alpha helix, which I hadn't said anything about, a long paper describing a whole lot of helical structures for alpha keratin, all of which were wrong, just because they were physicists. They didn't, if they had been chemists, they would have known about the theory of resonance, would have known that the peptide group in these polypeptides is planar, and that they had to include that restriction. They didn't. In all of their structures, the peptide groups were twisted around in a way that just isn't true, and it would have been rejected by the chemists. Well, I've come to the question, what is the nature of life? What is the basis of biological specificity? And that turning point that came for me in 1936, when Karl Landsteiner asked me how to explain biological specificity. How can the antibodies distinguish between hydrogen atoms around a benzene ring and a benzene ring that has a methyl group in the, say if it's benzoic acid, in the position meta or para to the carboxyl group? And that's something that we need to explain. Well, by 1940, I had formulated a theory of the specificity of serological reactions. And by 1948, my associates and I had verified this theory in many of its aspects. I'd kept asking myself, how can the antibody, using the intermolecular forces that we know about, van der Waals repulsion, if two atoms get too close together, they repel each other. The electron shells are involved in repulsion. Van der Waals attraction, a result of electric polarizability, their electrostatic interactions between these electrically polarizable atoms, and interaction of positive charges and negative charges, an ammonium ion group and a carboxylate ion group attract one another, hydrogen bond formation. These are weak forces that hold molecules or parts of molecules that aren't connected by covalent chemical bonds together. The conclusion that I reached is that there's only one way in which molecules can interact in this highly specific way, and that is through a detailed and close complementariness between the haptenic group of the antigen and the combining region of the antibody. I concluded also that precipitation by an antibody, agglutination and agglutination of cells, red cells, for example, by anti-erythrocyte antibodies, requires that the antibody be bivalent, have two combining groups. Here we have a big red cell, an anti-red cell antibody, homologous to haptenic groups on the surface of the red cell. The antibody has two combining regions. One grabs a haptenic group on this cell, and the other grabs a haptenic group on this cell. And so they hold the two cells together, and if there are a lot of antibodies, the red cells are clumped together, and you get the agglutinated red cells that, of course, cause trouble in blood transfusions. So the conclusion is that the antibody, that these two molecules interact with one another when they don't form real chemical bonds by as a result of the cooperation of a number of weak forces which cooperate over a surface area where one molecule fits very closely against the other. And this idea of a detailed and close molecular complementariness is, of course, similar to ideas that had been proposed before from time to time, going back, for example, to Ehrlich, lock and key sort of explanation, which means complementariness in structure. Well, but they weren't accepted in general. What do I have here? Oh, here's a list. Some of the people who worked during period about 1940 to 48 or 50 on these antibody studies. Dan Campbell, David Pressman, John Singer, now at UC San Diego, Art Pardee, we were talking about Art Pardee a couple of days ago, John Cushing at UC Santa Barbara, George Fagan at Stanford, Carlton Gajduszek, back in Bethesda, I guess, if he isn't in New Guinea. Here, I show Dan Campbell and two of our other associates in this work. My wife was a little bit irritated. We had a little old house on the front, or to one side on our place up in the hills above Pasadena, going back to 1860, and it was full of rabbit cages with rabbits that I injected with antigens every morning. And then we built a shed on the front lawn that contained the cages in which the breeding rabbits were kept to provide us with additional rabbits. She commented on this slide. She said, the only thing that slide shows is that you don't know how to hold a rabbit. Well, see, this sort of illustrates our idea of what an antibody. Of course, we know there are detailed structures of antibodies now with those long chains and short chains and sulfur disulfide bonds and I don't know what all. But I like to think of them as being like this early picture. And here would be an ovalbumin, anti-ovalbumin precipitate. The ovalbumin, I thought, it has molecular weight 43,000, antibody 160,000, and the ovalbumin, any part of its surface could have haptenic groups on it, and so it could have a large valence, and the antibody is restricted to small. And this predicts that you have 7 or 8 times as many antibody molecules as ovalbumin molecules, which, of course, is what's observed. Here's so we wanted to check this bivalent idea of bivalence. And we made these simple compounds which will precipitate with an anti-benzene arsenic acid antibody just the way that the azoprotein will precipitate with it. And all we did was to make the antibody against benzene arsenic acid, make the precipitate with one of these compounds, say either one of the two on the right side there that have two haptenic groups, and analyze it. It came out one antibody per di molecule, perhaps. And since the molecules have two haptenic groups, we said that the antibody must have two combining regions. Well, there was an immunologist who said he didn't think that we had proved that both of these were involved in the precipitation, both of the haptenic groups. So we made this compound. It has a benzene arsenic acid, R, on one side and a benzoic acid on the other side. And if you add this compound to antibody against benzene arsenic acid, you don't get a precipitate. If you add it to an antibody solution against benzoic acid, you don't get a precipitate. In each case, you get soluble complexes like this. But if you take antisera from two rabbits that have been injected differently, then the compound gives a precipitate with the mixture of the antibodies from the two rabbits, as shown here. You have an anti-R, and then R, and then X, the benzoic acid, anti-X, and then XR, anti-R, RX, and so on. I don't know how that antibody molecule with three combining regions got in there. It was put in by an artist, I think. Well, we determined hundreds of equilibrium constants between antibodies and haptenic groups, hundreds of them. This illustrates, this diagram shows some of the combining constants of anti-para-azobenzene arsenic acid, anti-meta-azobenzene arsenic acid, anti-ortho-azobenzene arsenic acid with a lot of substituted arsenic acids, and so on. And a lot of information. All I want to say about that is that there was a lot of information. And if the hapten, the molecule, was not rather similar to the one that had been added to the original immunizing antibody, it wouldn't combine very strongly. And since we knew the sizes and shapes of all of these simple molecules, we could say how close the fit is, just how big the pocket is in the antibody. And we decided that for benzene arsenic acid, the fit was to about a quarter of the diameter of an atom. Very close fit all around here. No doubt that you have this detailed molecular complementariness. In one case of para-azosuccinate ion, para-azobenzene succinate ion here, we were able, by using these equilibrium constants for a whole lot of substances, to show that the immunizing hepten had the succinic acid bent around into approximately a cis conformation, as indicated, and to show that there was a positive charge in the antibody right close to the carboxyl, the negatively charged oxygen there on the succinate ion, and that there was a hydrogen bond formed from between the antibody and the carbonyl group there, and so on, so that there was a great deal of information obtained. Well, I concluded that there was no doubt then that it was a detailed molecular complementariness in structure between the antibody and the antigen, the hepten, that was responsible for the specificity of combination there, and of course predicted that, or said that it was also responsible for the phenomenon of heredity. In fact, in 1940, well, here, jumping the gun a bit so I don't have to worry about the slide anymore, we have this remarkable example of complementariness with adenine and thymine forming 2 hydrogen bonds, guanine and cytosine forming 3 hydrogen bonds. probably the most important example of complementariness in structure in living organisms. So, in 1940, the German physicist Pascal Jordan, who had worked with Born on matrix mechanics, of course, knew physics, didn't know any chemistry, I'm sure, wrote a paper in which he said that the gene duplicates itself because, according to quantum mechanics, two identical molecules interact with one another more strongly than a molecule and a non-identical molecule. Max Delbruck met me on the campus and said that Jordan had published a paper, which I said was nonsense. So Delbruck and I published a paper saying that we had calculated the magnitude of the energy of interaction, and it was just completely negligible, this special quantum mechanical interaction between like molecules of the dimensions of molecules that are present in the human body, and that, in fact, the gene consists of two mutually complementary strands, and that the gene duplicates itself by the unfolding of the pair of mutually complementary strands so that each can act as a template for the synthesis of the other. Well, later on, Watson and Crick, that was 1940 that we published that paper. Many years later, Watson and Crick discovered the double helix with the help of Jerry Donahue, who pointed out that they were using the wrong structures for the nitrogen bases, that the correct structures could form hydrogen bonds with one another. That was a valuable contribution. He's the only person whom they thank in their paper on the double helix. They could have thanked him even much more strongly, I think, than they did. So we have that example of complementariness. Well, even back in 1940, I was feeling that I didn't need to worry much any longer about what the secret of life is. It's molecular complementariness. And of course, after Watson and Crick, I was nearly completely satisfied. If I were giving another lecture, I could go on in detail. We can imagine how the primitive Earth was, and there developed the hot, thin soup of oparin and haldane as ultraviolet and light and lightning caused all sorts of chemical reactions to go on. Every conceivable kind of moderately simple molecule to form. And with the situation far from equilibrium, a catalyst could cause one reaction to go much faster than others so that you would get a preponderant number of molecules of a certain sort. Adenine might speed up the production of thymine, and thymine speed up the production of adenine. Enzymes, polypeptide chains, polynucleotides would come into existence, and single-celled organisms, and we could have rapid reproduction and billions and billions of cells mutating and competing and for 2 billion years, building up finally the extremely complex system of living organisms that we have on Earth now. And so I'm happy, even if I don't follow all of the details as the biochemists and others, molecular biologists, are working out now. I'm happy to feel that I have, and have had for a long time, an understanding of at least the basic answers to the question, what is life? Well, I've taken pleasure in being here in this great university and having contact with scientists who are learning more and more about the nature of life. Not the basic question, that's been answered, but the frills. I might make a remark about the double helix. My wife once said to me if that was such an important problem, why didn't you work harder at it? And, well, I shan't try to answer the question. That's just an introduction to something else that she said. She said, the most important of all activities is that of keeping the world from being destroyed in the nuclear war. So I conclude by saying that the most important of all problems is that of keeping the world from being destroyed in the nuclear war. And also to say that every one of you should do whatever you can to prevent this ultimate catastrophe.
Well, I'm going to take your microphone a minute just to thank you for that. And I should sit there then. I'll give you one more chance. I think we have to end. It's very sad for all of us that we have to end these lectures, but I'd like to thank the Hitchcock Committee for making such an excellent choice, and Dr. Linus Pauling for giving us such an honor on our campus to spend this time with us. Dr. Pauling has not only given 2 lectures, but has met with students, faculty, in almost a continual round of, I think, exploitation of a visitor. I don't think we've given him a moment's rest since he's been here. I think one of the great contributions of the Hitchcock lectureship is something that gives us an extra dimension beyond the articles. I think although we reviewed Dr. Pauling's many accomplishments, most of you have read them in textbooks, in articles. I think his personal magnetism and his excitement of what he's given us to this campus during this week has illustrated, I think, 2 features that have characterized his career. One is courage both in political area and the courage to take new scientific ideas. And the second is his enormous enthusiasm for life and for intellectual ideas. In fact, I looked back over the history of the Hitchcock professorship and found that in many cases it was a recruiting device, that some of the people who were Hitchcock lecturers ended back in Berkeley. And so if I had to pick an able young assistant professor who's shown some ability, who had enormous enthusiasm and many years of productive ideas ahead of him, I would nominate Linus Pauling. It's been a great pleasure to have you here.